Welcome to series of lectures on digestive system. I am the senior lecturer of Department of Histology, Embryology and Cytology of Azerbaijan Medical University, Dr. Əyyubova. Today I am going to talk you about the initial part of the digestive system, that is the oral cavity. Here we will focus on differentiation of parts of primitive gut tube during embryogenesis, morphofunctional characteristics, and general plan of structural organization of oral cavity, teeth and the development, the role of primitive oral cavity and surrounding its structures in development of phase of the embryo. The digestive system is composed of the oral cavity, alimentary canal, which include esophagus, stomach, small and large intestine, and the anus. Also associated glands, major salivary glands, liver, and pancreas, which also associated with alimentary canal. The main function of the alimentary canal, the digestive system, is ingestion, mastication, swallowing, digestion, and absorption of food substances, and also it will eliminate the undigestible remnants. So in broader aspect, the main function of the digestive system, it's broken down the proteins, lipids, carbohydrates, and these substances will broken down into small units that can be absorbed by the small intestine. The inner surface of the alimentary canal is critical barrier between the outside of the body and its contents of the lumen of the body, and its structure reflects its functions. Let's give a brief review on the development of various parts of the digestive system. As we know, in embryonic life, let's look here. If you remember, we had the embryonic disc here, which initially has, the embryo has a disc shape. Then above the disc we have amniotic cavity, and below it we have yolk sac cavity. And the gastrulation created three layers of the germ disc: ectoderm, mesoderm, and the endoderm. We know that as a result of cephalocaudal, let's look here, cephalocaudal folding, and also lateral folding of the embryo, a portion of the endoderm derived from gastrulation, here the yellow color, that is the endoderm. It is incorporated into the embryo to form the primitive gut. Let's look here, the formation of the primitive gut that is lined by endoderm germ layer, and the yolk sac cavity together with allantois will remain outside of the embryo, so the embryo body is developing here. In the cephalic and caudal parts of the embryo, the primitive gut forms a blind ending tube, the foregut and the hindgut. Between them we can see the midgut. It remains temporarily connected to the yolk sac by means of the vitelline duct. Let's look here, the vitelline duct, that yolk sac, and the primitive gut tube. Here we can see. Development of the primitive gut and its derivatives is usually, we will take in part for four sections. That will be pharyngeal gut, here, pharyngeal gut, and or pharynx, that extends from the oropharyngeal membrane or buccopharyngeal membrane, that is ectodermal endodermal thickening, till the respiratory diverticulum here, where we'll develop the respiratory system, that tracheobronchial diverticulum, it will give rise to the respiratory system. So from the oropharyngeal membrane till the tracheobronchial diverticulum, we will have pharyngeal gut or the pharynx. Then we will have foregut, which will locate it between the respiratory diverticulum till the liver bud, where we'll develop the liver. So that will be the foregut. Then we will have midgut. Midgut is located between the liver bud and the region is in between the right 2/3 of the transverse colon and the right and the left 1/3 of it. It will be the region of the boundary for the midgut and the hindgut. Then we will have hindgut, which will extend till the cloacal membrane. Here we have to take in mind that endoderm, which is colored by yellow color, this tube, the endoderm will form epithelial lining of the digestive tract, and also it will give rise to the specific cells of the glands, such as hepatocytes and also exocrine and endocrine cells of the pancreas. The stroma, the connective tissue of these glands, will develop from the visceral layer of the lateral mesoderm. The mesenchyme for formation of the neck and head region, it will derive from paraxial, lateral plate mesoderm, neural crest cells, and from the ectodermal placodes. The most distinctive feature in the development of the head and neck of the embryo is the presence of pharyngeal pouches. These pouches appear in the 4th and 5th weeks of development along the lateral walls of the pharynx. Here we can see the formation of, first of all, arches, then they will develop into the pouches. So pharyngeal pouches. 5 pouches we will see in the wall of the pharynx. They play important role in the development of neck and the face of the embryo. At the 4th week of development, in the face of the embryo, we can see 5 outgrowths, protuberances, prominences. They are frontonasal prominence, it is unpaired. Then paired mandibular arches. The mandibular arches form the lowest inferior wall, and that is the first pharyngeal pouch. Then we can see in the lateral sides we can see maxillar prominences, that is the dorsal portion of first pharyngeal pouch, and so that is the frontonasal prominence. So 5 prominences we can see here, and that is the oropharyngeal membrane. If you remember, that was ectodermal, endodermal thickening. Then we will see appearance of nasal. The development of the face will be continued by the formation of the nasal protuberance, which will detach from the frontonasal prominence. Differentiation of all of these structures will depend on the epithelial mesenchymal interactions that we have to take in mind. So frontonasal prominence then will give rise to forehead, nose, primary palate, nasal septum, and philtrum of the upper lip. But maxillar prominence will develop into cheeks, maxillar bone, secondary palate, and lateral portions of upper lip. Mandibular prominences will develop into low jaw and the lip. So the face of the embryo will develop. What we can see here, if you remember, that are lens placodes, that is the region where the eyes are going to develop. The oral cavity. Today we will focus on the initial part of the alimentary canal, that is the oral cavity, and we will go into details of its histological structure. As students of dentistry faculty, we have to know the details of histological structure or the components which form the wall of the oral cavity. So the oral cavity, it is the initial part of the alimentary canal, and its function in the receiving and initial processing of food, and so we will cover all of the components of the oral cavity. the oral cavity that is entrance to the digestive system, as such, is the primary barrier for the pathogens. Here taking place ingestion, mastication, then the partial digestion, mechanical and chemical. Then there taking place lubrication of food into small spherical masses known as bolus. And it is actually we have to know that that is not organized in 4 layers as the rest of the digestive tube. Here we will distinguish the tongue, then we will focus here on the structure of the lips, then palates, then cheeks we are looking here also, and finally we will discuss teeth. The oral cavity actually it is lined by the oral mucosa. The oral mucosa we have of 3 types. First of all, that is the let's talk about the masticatory mucosa. Masticatory mucosa covers the regions which mainly undergo some friction and mastication. That is the hard palate, dorsal surface of the tongue, and also gingiva in the where the tooth are resting. So that is the masticatory mucosa. It will be covered by stratified squamous keratinized to parakeratinized epithelium. The rest of the oral cavity covered by the lining mucosa that is covered by non-keratinized epithelium. Then the specialized mucosa we will have on the regions where we can find taste buds. The taste buds that are specialized regions for taste discrimination. How we can distinguish the keratinized, parakeratinized epithelium? Let's look here. First of all, let's look at the non-keratinized epithelium, that is non-keratinized, stratified, squamous epithelium, several layers of epithelial cells we can distinguish here. But pay attention that all of these cells in all of the layers, they include nuclei. It means that this is non-keratinized epithelium that covers most part of the oral cavity. Also, it will cover the esophagus, it will cover the pharynx. Now let's look at the parakeratinized epithelium. In parakeratinized epithelium, the surface layer, in surface layer, some of the cells include nuclei, but most of them do not include nuclei, so they are dead cells. But in keratinized epithelium, as that is the same as structure of the epidermis, that is keratinized epithelium, it means that surface layer of cells do not include any nuclei. That's taking place keratinization here. Let's focus on the lips. Lips form boundaries, anterior boundaries of the oral cavity, and their main bulk, that is their main part, is formed by skeletal muscle, which form the bulk, the central portion of the lips. That is actually, that is what is the muscle here, and that is skeletal muscle. Then lips covered by depending on the region. First of all, it has, that is first region here, that is external aspect, that is skin portion, the sagittal section of the lip here, that is section that is stained by methoxylin staining. So outer aspect of the lip is covered by skin, thin skin. It includes hair follicles, sebaceous glands, sweat glands. Then we are coming to vermilion zone. Vermilion zone, that is pink region of the lip, that is also covered by thin skin, but unlike the skin portion, they are absent. We cannot find sweat glands and hair follicles. Also, we can find here sebaceous glands, but they are non-functional. It means that there are not any function. The absence of functional glands in the vermilion zone necessitates the occasional moistening our lips by the saliva. What is the other feature of the vermilion zone? It is the skin here, epidermis, especially its surface layer is so very thin, and the dermal papilla too tall, and they reach in capillary network. That's why the red apparatus between the dermis and the epidermis is well developed. So that the capillary loops of the dermal papilla are close to the surface of the skin, imparting a pink color to the vermilion zone. Then we are coming to the mucous aspect. The mucous aspect, it faces the oral cavity, and it is covered by stratified squamous non-keratinized epithelium. The subepithelial connective tissue is that is lamina propria of the mucosa. It's composed of the dense irregular connective tissue that contain glands. These are the minor salivary glands that produce saliva into the oral cavity. Then we are coming to palate. The palates, we know that they form the roof of oral cavity. Let's look here. We can distinguish the hard palate, then the soft palate. The hard palate, as we just mentioned, covered by masticatory mucosa. It means that here the epithelium stratified, keratinized, and under the epithelium we can distinguish the dense irregular connective tissue which forms the lamina propria of mucosa of hard palate. And here in the connective tissue in anterior portion of the hard palate, it includes a little amount of adipose tissue, but in the posterior aspect it may contain minor salivary glands. So the lamina propria of mucosa is tightly adhered to the bony shelf of the hard palate. Then we are coming to the soft palate. Soft palate, as in hard palate, it's you know that it separates the oral cavity. Here below we can see oral cavity, and above that is nasal cavity. So it separates these two cavities from each other. So the face of the soft palate that faces the nasal cavity, it will covered by respiratory epithelium because it faces the nasal cavity. It is pseudostratified ciliated epithelium, but the face that faces the oral cavity, it is covered by stratified non-keratinized stratified epithelium. Under the epithelium we can distinguish the lamina propria that contains also the small salivary glands. Actually, it is also composed of the dense, irregular connective tissue. Now let's go to next component of the oral cavity. That is the largest component. That is the tongue. That is it has extreme mobility, and it participates not just in digestion, in mastication, but also formation of the speech. And it has dorsal surface and ventral surface. The dorsal surface is covered by masticatory mucosa, and the regions where we will have taste buds, that will be specialized mucosa that is specialized for taste perception. If we will look at its detailed structure here, we can distinguish that it has anterior two portions and posterior portion. Between the anterior 2/3 and posterior 1/3 is located sulcus terminalis that at the apex has foramen secum. Here we also can see the root of the tongue where we can distinguish the lingual tonsil. That's why the surface of this part is very rough. On the dorsal surface of the tongue we can distinguish the lingual papilla. Lingual papillae, they are extensions of the lamina propria into the epithelium. Epithelium here, depending on the type of papillae, it may be stratified, keratinized, for example, in filiform papilla, which are mostly abundant, and scattered throughout the surface, dorsal surface of the tongue. They are filiform papilla, and they do not include taste buds, so they do not participate in taste perception. The fungiform papilla, which are mushroom-like papillae, but on their surface we will have taste buds on their lateral surfaces or on the apical surface. The foliate papilla located along the dorsolateral surfaces of the tongue, and they are rudimentary for adult human. Here we are looking at the taste buds. Pay attention, taste buds, which participate in taste perception. We studied them in organs of scent. That's why I will not focus on the structure. We are looking here at the filiform papilla, then foliate, pay attention, and finally we are coming to the circumvallate papilla. Circumvallate papilla, they located in front of the sulcus terminalis in the V-shaped fascian. These papillae are submerged into the surface of the tongue so that they are surrounded by epithelially lined groove. Pay attention to this groove here. And this base is pierced by slender ducts of the von Ebner's glands. Pay attention to Sirot's glands here. They are the von Ebner's glands. They are also types of minor salivary glands, and their ducts open into this groove around the circumvallate papilla. The epithelial lining of the groove and the side of this papilla have taste buds. So under the epithelium we can see the lamina propria, connective tissue of the lamina propria, ducts of glands, these are small minor salivary glands, and striated muscle. As we know, the tongue, it is a mobile organ because it has two groups of muscles. There are two categories. The extrinsic muscles originate outside but insert into the tongue and move it. Intrinsic muscles are contained wholly within the tongue and alter their shape. Palatine tonsil. Palatine tonsil, it is part of the lymphoid system that is protect the immune system that protects our body from any invaders. So the palatine tonsils, these are part of lymphoepithelial ring or Waldeyer ring that surround the entrance of the alimentary canal and also the respiratory system. The palatine tonsils, they are bilateral and they are located at the boundary of the oral cavity and the oral pharynx between the palatoglossal and palatopharyngeal folds. The deep aspect of each palatine tonsil is isolated from the surrounding connective tissue by the dense connective tissue. It is submucosa actually of the pharynx which surround the region of the tonsil. In large magnification we can see here that submucosa which surround, and it forms capsule of this organ. But inside of the palatine tonsil we can distinguish lymphoid nodules, individual nodules. Here they have germinal centers. These are regions for proliferation and differentiation of the lymphocytes, mainly B lymphocytes. Superficial aspect of the tonsil is covered by stratified squamous non-keratinized epithelium, that is non-keratinized epithelium, and that dips into the 10 to 12 crypts. These are invaginations, and these invaginate into the tonsillar parenchyma. The crypts frequently contain food debris, desquamated epithelial cells, dead leukocytes, bacteria, and other antigenic substances. The parenchyma of tonsil is composed of the numerous lymphocytes, and here taking place the proliferation of lymphocyte T and B lymphocytes. Here we are looking also the tonsil, that is epithelium which covers the lymphoid follicles, and that is a crypt. Pay attention, it is invagination of the epithelium, that is crypt. These crypts will, of course, they will increase the surface area of it. Let's now go to the structure of the tooth. We know that humans have two sets of teeth, the 20 deciduous teeth, which are replaced by 32 permanent teeth composed of the 20 succedaneous teeth and 12 molars. Here we are looking that each of the tooth, it has its own morphological features, and also it will have its own number of roots, so they are different, and here you can see which types of tooth we have in our oral cavity. Each tooth is suspended within the alveolar bony socket by the periodontal ligament that maintains the tooth in its alveolar bony socket. The gingiva also supports the tooth, and its epithelium seals the oral cavity. Let's look here. The epithelium of the gingiva will seal the oral cavity and from the subepithelial connective tissue spaces. The portion of the tooth that is visible in the oral cavity, it is called clinical crown. But we know that there is also anatomical crown. What is the difference between them? Pay attention to the root of the tooth, which is located inside of the bony socket. Between the root and the crown is located the neck of the tooth. That region which comes after the neck, that is anatomical crown of the tooth. But clinical crown, that is that part which is seen in the oral cavity. It means that some part, the partially enamellum covered by gingiva, and it will cover partially the crown of the tooth. Inside of the tooth, we are looking at the cavity which is called pulp chamber and root channel. Inside of them, we can see the pulp, that is the soft tissue of the tooth. Then we are looking at the mineralized hard tissues of the tooth. Hard tissues represented by enamellum, which covers the crown, then dentinum that surround the pulp chamber and root channel, and then cementum. Cementum will cover the dentinum. So the bulk of the tooth is composed of the dentinum. Enamellum and dentinum meet each other at the cervix of the tooth. Let's look here. Also, periodontal ligament will, here you can see the periodontal ligament. The root canal communicates with this periodontal ligament at the apical foramen. It is at the tip of each tooth. Here, through this opening, the blood vessels and lymphatic vessels, also nerves, enter into the tooth. So now let's talk about the enamellum. Enamellum, it is the hardest tissue of the human body. It is composed of mainly at the 96-97% of inorganic substances, and also it includes organic substances, which are represented by the specific proteins that are enamellins, amelogenins, and ameloblastins. Enamel is produced by cells which called ameloblasts. Let's look here. There are prismatic shaped cells. In embryonic life, they produce enamellum. That is the enamellum which is produced in the embryonic life. The enameloblasts produce enamellum in segments, and these are rod segments. It's approximately each day they produce 4 to 8 micrometer segments. Then these successive rod segments adhere to each other, to one another, and form the cylindrical shaped enamel rods. Pay attention. So each enamel rod is produced by one ameloblast. When the cell produces this enamellum, they go away from the surface, from the dentinal junction, enamel dentinal junction, they go away and they form enamel rods. Each enamel rod will have a shape like a keyhole shape. It will have cylindrical head and the tail region, which is less wide, it's narrow region. And then enamel is non-vital substance. We have to understand and remember that it is because amyloblasts die before the tooth erupts into the oral cavity, so the body never can repair the enamellum because these cells die during eruption of the tooth. And because during its formation enamel is elaborated in daily segments, the quality of enamellum will depend on the health of the mother during prenatal stage of or on the health of the adult person after birth. In childhood, of course, the enamel rod then reflects the metabolic state of the mother during the time of enamel formation, and it results in successive rod segment sequence. And here we will see normally calcified regions and with hypocalcified regions, and it will be reflected on the surface of the enamellum, and it will see like lines of reticules. Histologically, it will appear like a stria of reticules. And then on the free surface of the newly erupted tooth, we will be able to see the cuticle, that is primary enamel cuticle. Then it will wear off in initial mastications. That is like basal lamina like structure. Here we are looking at these enamel rods. They have the head region, the tail region, as we can see here. As we know, they are elaborated by enameloblasts. The second hard tissue of the tooth, it is dentinum. And what is the dentinum? Pay attention here. Here the amount of inorganic substances is less than in enamellum. The 65 to 70% of dentinum is composed of inorganic substances, 25% approximately that are the organic substances, which are represented by collagen first type, then glycoproteins, proteoglycans, and also in dentin we can, the 10% of it represented by the water. About the structural organization of dentinum, we should know that it is elaborated by the cells odontoblasts, which are located at the periphery of the pulp. And these odontoblastic cells, they are processes located inside of the dentinal tubules, which extend from the pulp towards the dentinal enamel junction or the dentinocementum junction. So they radiate throughout the length, throughout the wideness of the dentinum. Then what we have to know that odontoblasts, they are living cells throughout their life. That's why they will provide reparation of the dentinum throughout the life, and unlike the amyloblasts which die, that before eruption into or tooth into the oral cavity. And also we have to know that as in enamellum, the dentinum also is produced in daily segments, 4 to 8 micrometer in segments. That's why the segments will represent the healthy state of the mother in the body of which develops the embryo. That's why these regions of hypocalcified and normally calcified segments will be represented by the histologically will be represented by lines of Owen, which are similar to the lines of reticules on the surface of enamelum. Then what we have to know about the hard tissues of the tooth, that is a cementum. Cementum covers the root of the tooth, and it is similar to the bone tissue, in which here also 50 to 55% is inorganic substances. Then approximately 50% here we will find the organic substances, which are represented by collagen fibers. These are first, third, 12th type of collagen, also glycoproteins and proteoglycans. In cementum, we can find the regions which reach in cells, that is cellular cementum, that are regions of near to the apex of the root, where we can distinguish these individual cells, and acellular cementum, which is near to the neck regions of the tooth. It doesn't matter. All of the surface of the cementum is covered by cementoblast cells, that cells which produce cementum, and that they cover all of the surface of cementum. So the cementocytes located within the lacunes, and their processes located within the canaliculae. These canaliculae will contact the blood vessels in periodontal ligament. Also, from periodontal ligament, the collagen fibers will insert into the cementum. These are those collagen fibers which maintain the tooth and attach the tooth to its alveolar bony socket. These fibers, where they attach to them, insert into the cementum, and they're in regions where they insert into the alveolar bone. In those regions, they will called Sharpey's fibers. They are mainly represented by the collagen first type, and the collagen fibers are produced by fibroblasts, which are present here, and these fibroblasts responsible for high turnover of the collagen. You know that in all of the body, the region where collagen fibers undergo a high rate of turnover to renewal, that is a periodontal ligament, and that's why when we have deficiency of vitamin C, it's firstly will be seen in the gingiva, in gums. They become soft, there we will see bleeding, and also even the tooth will loosen in the place, and even we can lose the tooth. That is a result of problems in collagen fibers, because as you know, collagen fibers need for proper production of collagen fibers, we need vitamin C. That's why deficiency of vitamin C is mainly the first signs of this deficiency we will see in our gums, gingiva, and in periodontal ligament. Then when we told about the hard tissues of the tooth, now let's come to soft tissues. About first of all, periodontal ligament, it's also belongs to soft tissue. Now about the pulp. Pulp is located, as we know, in the lumen of the tooth, and that is the soft, the collagenous connective tissue, loose connective tissue, rich in connective tissue cells, fibers, ground substance, as in other places of the body. But what is special for the pulp, it is the morphological appearance of it. The pulp we can subdivide into odontoblastic zone, where located odontoblasts, that is peripheral part of the pulp. Under the odontoblastic zone, we have cell-free zone, that is devoid of cells, and under of it we will have cell-rich zone, that is rich in the connective tissue cells. That zone will surround the pulp core. The core of the pulp include high amount of the blood capillaries, the nerve fibers. The nerve fibers are of two types. Sympathetic fibers will control the lumen of the blood vessels, and here we also have pain fibers, which are responsible for pain sensation, and they form here the Rashkow nerve plexus. And as you know, all of them enter the pulp through the apical foramen. Then Let's go to the odontogenesis, that is the development of the tooth. How the odontogenesis begins? It starts at the 6th to 7th week of embryonic development, and it starts with formation. Actually, what are the structures which participate in the tooth development? That will be first ectoderm, which line the oral cavity, then the mesenchyme, which located under, and that mesenchyme derived from neural crest cells. That's why it's called neuromesenchyme. In the oral cavity, which is just one cavity where without vestibulum or the oral cavity proper, at that region there develops two horseshoe-shaped bands of cells. These are the maxillar, along the maxillar arch and mandibular arch. That is a band of cells. They will also can we can call the dental lamina. Pay attention to the illustration. In cross section we will see like this, and from them we will have the next step. There, these horseshoe shaped laminas will have subdivided into two components. First of all, the vestibular lamina, which will separate the vestibulum of the oral cavity from the oral cavity proper. Then it will have another part, that is dental lamina proper, from which it will develop the tooth. From the dental lamina, in the 7th week, in 8th week of gestation, will develop extensions. They will called buds as a result of division of these cells. And remember that is an ectoderm germ layer, derivative of the ectoderm germ layer. It will form buds. 10 buds we will see along each of these arch, 10 buds along the mandibular arch, and 10 buds along the maxillar arch. These buds, now let's come to the main stages of tooth development. From those dental lamina, as we know, now starts growing the buds, and it will tell us about the bud stage, that is initial stage of tooth development. Bud, as we know, it is developed from the ectoderm germ layer, and that is band of cells which penetrate into the underlying mesenchyme. Then from the buds, the result of development or differentiation of its cell, division of these cells will create the enamellum organ. That's enamel organ, or it will have shape like cup, and that's why this stage will called cap stage. In cap stage, the enamel organ, if we will look at its structure, we can distinguish here the concave side, that is called inner enamel epithelium, and the convex side, that is outer enamel epithelium. Between them located stellate reticulum. Under the influence of this enamel organ, the underlying mesenchyme also undergo condensation. The cells undergo condensation, and here they will form dental papilla. So that the enamel organ, together with this condensed mesenchyme, which now we call dental papilla, will form tooth germ. Now we are going to next stage, that is bell stage. At this stage, it's like bell. The structure will have shape like bell. And what does it mean? Here in bell stage, we will see the appearance of new cell mass under the inner enamel epithelium, and this one will called stratum intermedium. The stratum intermedium will simulate the cells of inner enamel epithelium to differentiate into amelloblasts. Then the outer enamel epithelium begin to degenerate, and then what we will see, all of this structure, the enamel organ together with dental papilla, it is surrounded by mesenchyme. These mesenchymal cells also condense, accumulate here, and form band of cells that surround both of these structures. That one will called dental sac. So the dental sac will surround both of these structures. Here I want to tell you something that is very important. It is a process of morphodifferentiation. So the Bell stage is talk us about the morphodifferentiation and histodifferentiation of the tissues of the tooth. And here we have to know that this is responsible for establishment of the template of the presumptive tooth. That is, the enamel organ assumes the shape of incisor or canine or molar. This event is controlled by primary enamel knot. It is dense clump of cells located adjacent to the inner enamel epithelium. So adjacent to inner enamel epithelium, we will have enamel knot. It is dense clump of cells, and it is inside of inner enamel epithelium within the substance of enamel organ. And it appears that the ectomesenchymal cells of the dental papilla, which is located on them, they produce some cytokines, the signaling molecules, which will induce enamel knot, and it will become the principal signaling center of tooth morphogenesis. Cells of primary enamel knot synthesize bone morphogenic proteins, also fibroblast growth factor. And it will be produced in particular relationship, sequential relationship that result in cusp formation in developing tooth. And to be able to establish this time-specific relationship, the ectomesenchymal cells of dental papilla will produce epidermal growth factor, then fibroblast growth factor. And the primary enamel knot cells without these substances will go to death, will die by apoptosis, and cusps will not develop. When the cusp formation in responsibility to signaling molecules will start, then the cells of enamel knot will move towards that cusps, those cusps, and in their place will develop secondary enamel knots, and so the future cusps will develop. And when the primary and secondary enamel knots have performed their functions, the ectomesenchymal cells will finish their release of epidermal growth factors and fibroblast growth factors, and the presence of bone morphogenic protein drives the cells of the primary and secondary enamel knots into death, into the apoptosis. So the dental papilla, whose most peripheral layer of cells is separated from inner enamel epithelium by the basal lamina, is responsible for the formation of the pulp and dentinum of the tooth. And now we are talking about the continuation of the Bell stage. And now we know that inner enamel epithelium will develop into the amyloblasts. The amyloblasts will begin to produce enamellum. Enamellum will be produced in downward direction. And under the influence of these cells, the most peripheral cells of dental papilla, which is here we know, these cells will begin to produce dentinum. So pay attention that dentinum is also produced. So that peripheral cells of the dental papilla will develop into the odontoblasts, and odontoblasts will produce dentinum in upward direction. So if ameloblast produces enamellum in downward direction, that odontoblasts produce dentinum in opposite direction, so upward, and so the dentinum and growing enamellum will meet each other, and they will establish dentino-enamellum junction, and this stage will called oppositional growth stage. Now let's look here what is the oppositional growth stage. First of all, let's look at our cells that are odontoblasts, then amyloblasts. Odontoblasts produce dentinum, amyloblasts produce enamellum, and they meet each other at this junction. That's why it's oppositional growth stage. Here we are looking in the example of our tooth, and that is the hematoxylin eosin staining slide, histological slide. And let's look at the same structures here. The stellate reticulum, the stellate reticulum, it is the pulp of the enamellum organ. Then stratum intermedium, pay attention, stratum intermedium here. Then we can distinguish here the amyloblast cells, pay attention, amyloblasts, which are in secretor state, that is written here. You can see amyloblast cells. Then here will be enamellum that is produced by these amyloblasts, that is a result of activity, secretor activity of amyloblasts. Then let's look at this side, the left side of illustration, that is dental pulp, dental papilla, we know it, and we know that peripheral cells of dental papilla develop into odontoblasts. They produce dentinum, pay attention, that is predentin, then dentin. What is the difference between them? Predentin, it is immature dentin, so in first stage the odontoblasts produce predentin. The predentin, it is the dentin, but without mineralization, so there are not deposited calcium hydroxyapatite crystals yet. Then it will be converted into dentinum when there will be taking place mineralization. So dentinum and enamellum. So the enamel dentinal junction here. We pay attention now, odontoblast cells and dentinum, then predentinum. Here that is a dental pulp actually. Dental pulp we are looking here. And now we are coming to the differentiation of the layers of the tooth. Now let's look at the development of tooth in our slides. That is first stage of tooth development, that is enamel organ, that is inner epithelium, outer epithelium, between them stellate reticulum. Here we are looking at the dental papilla and the dental sac, which surround both of these tooth germ structures. And here the late stage, here we can see the dentinum, enamellum, and we will emphasize some points here. We know that peripheral cells of dental papilla develop into odontoblasts that produce dentinum. The cells which are located inside of the dental papilla, here is written dental papilla, these cells develop into the pulp of the tooth, so they give rise to tooth pulp. Then the outer enamellum epithelium will degenerate, but the stellate reticulum will develop into the cuticule which will cover the tooth. And what will develop from the dental sac? The innermost cells of dental sac will develop into cementum, and outer cells of dental sac will develop into the periodontal ligament. So we know now that from all of these structures that I mentioned today, just enamellum organ has ectodermal origin. And now we know that from enamel organ develops just enamellum of the tooth and the cuticle, which the membrane-like structure, which then will wear off during the first mastication. But all of the components, all of other components of the tooth will develop from the ectomesenchyme, which were underlying the ectoderm. So that is ectomesenchyme will give rise to all of the remainder tissues of the tooth. Here we are looking at dental pulp is developing, and from which source it developed. Now we know that it's from the innermost cells of the dental papilla. Then when the crown of the tooth is developed, now starts development its root root of the tooth, and that is root formation. And this stage, the outer and inner enamel epithelium, which form cervical loop, they elongate and form two-layered sleeve-like structure known as Hertwig root sheath. Pay attention, it will be a sleeve-like structure which is called Hertwig epithelial root sheath, which encompasses ectomesenchymal cells located deep to the developing crown, forming an elongation of the dental papilla. Pay attention, they form here the root sheath. In the root sheath then will form perforations, and that cells will degenerate so that the cells of the dental sac will penetrate through them, and they will establish contact with the odontoblast layer, with the dentinum actually, and they will form here cementum. So the dental sac cells, innermost cells, will develop into cementum. The remainder cells will form periodontal ligament. And the elongation of the root is a consequence of the lengthening of the Hertwig epithelial root sheath. That is a very important point which we have to remember. As the root becomes longer, the crown approaches on the and eventually erupts into the oral cavity. And the lengthening of the root is not responsible for tooth eruption. Instead, the two processes merely happen to occur simultaneously. So it means that not root elongation is responsible for eruption of the tooth. Then let's go to next slide, periodontal ligament. Pay attention, and it is located in the periodontal ligament space, defined as a region between the cementum of the root and the bony alveolus. This richly vascularized connective tissue has principal fiber groups and the sharpest fibers. As we know, they insert into the alveolus and the cementum. And something about the gingiva. We know that pay attention, that is the tooth, and that is gingiva, the gum, pregingival surface group we can see here. And what we have to know from here, that the region of the gingival epithelium that attached to the enamel surface here, to the enamel surface, and it is known as junctional epithelium, and it forms collar around the neck of the tooth. The junctional epithelium forms a barrier between the bacteria-laden oral cavity and the sterile environment of the gingival connective tissue. So it will isolate the connective tissue from the oral cavity, which reach in the bacteria and everything. Here we are looking at two cells, that is an ameloblast cell, that is the odontoblast cell. Ameloblast cells will have Thom's process, that is result of elongation, that when the ameloblasts move away from the tooth surface, that process undergo elongation. And then the odontoblast cells, you know that they have processes, and they located inside of dentinal tubules. So that is the end of the lecture. Thank you for attention, and we will continue the series of digestive system lectures, and you will have 2 additional lectures on this topic.